morning, I'm Sophie Rovner. Welcome to this news briefing from the 253rd National Meeting and Exposition of the American Chemical Society in San Francisco. We're joined today by Dr. Daniel Nocera from Harvard University. He will be talking to us about his work on a bionic leaf that could help feed the world. Dr. Nocera.
Hi. We just completed a study. It's not published yet for the press because people have been asking me for the paper. So it's very dangerous to talk about science that you haven't published, but this is complicated enough. I don't think we're going to get scooped anytime soon. And we'll be, just so for the press understands, we'll be submitting that paper within the next month to 6 weeks. So I'll send copies when it's written up and submitted. But what we did is, and I should start earlier in the year, we published a paper in Science where we took an engineered microbe that only eats hydrogen. That's its only food source. And then it can breathe in CO2, and it made liquid fuels. And the hydrogen came from solar water splitting. So the thought process is sunlight splits water and makes hydrogen and oxygen. I've then bottled the sunlight in hydrogen. I've basically taken the sunlight and converted it to a fuel, hydrogen. Then we took an organism that was engineered to eat the hydrogen as its only food source, so it's kind of eating the sun then. And then once it ate the hydrogen, it could convert it to NADPH, which goes to ATP, and that's cellular energy. So we're able to basically take sunlight to cellular energy through water splitting. And then once it had that cellular energy, it could power a cycle to take some more hydrogen plus carbon dioxide and make liquid fuel. After that, we said, what else is in the air? Of course, nitrogen. And so what we did is the same type of process. We still did hydrogen plus CO2 to fuel, but we didn't make a liquid fuel that the organism expelled. It kept the fuel inside, and it kept it inside as polyhydroxybutyrate, which is a bioplastic. So now the bug is taking H2 plus carbon dioxide to bioplastic, and that's an internal fuel supply for the bug. And this bug also has nitrogenases, an enzyme which can take nitrogen from the atmosphere. It's taking hydrogen in, and it can then take the nitrogen plus hydrogen to make ammonia. So this bug's doing two things with hydrogen. It's breathing in carbon dioxide and making bioplastic, and it stores it in the cell as its fuel supply. And then another part of the bug is taking nitrogen plus hydrogen and making ammonia. Now, NH3 usually gets forward propagated to proteins and all sorts of needs that you need nitrogen amino acids in your body. So what we do is put a small molecule block in and let the ammonia start building up, and then this bug literally just starts expelling ammonia. Because it has its own energy supply, it doesn't need to see the sun anymore or hydrogen. It already took care of that, making bioplastic. We can then bury it in the ground, and we can grow big radishes, like really big radishes. So it's literally, we can grow food in your backyard. And so basically what we've done is Haber Bosch, but I don't need a big plant, and I don't need a big distribution system, and you can do it in your backyard, all powered by the sun. So that's what the work is about, and what I'll be talking about later today, and then publishing in the next, hopefully, 6 weeks, or submitting for publication. So I think that's a good summary.
All right, thank you. Are there any questions? And if so, could you please wait for the microphone, state your name and affiliation before asking your question.
Sam Lemonick, freelance.
Hi, Sam.
How are you? Can you talk a little bit more about the block, the small molecule block that causes the ammonia to accumulate?
It's a small molecule. It's called a glufosinate. It's a small molecule actually used in the agricultural industry also, actually to kill things. Because if I want to kill a weed, I actually don't want it to make proteins, and I don't want it to do everything I just said. And so we put a little bit of that in, not so much that we're going to kill the organism. And as a matter of fact, the organism keeps doing fertilization as long as it has polyhydroxybutyrate, its internal fuel supply. Once that's used up, it's dead because it doesn't have any more energy supply anyways. So it's a balancing act. You put the small molecule block in not to kill it, but allow it to make enough ammonia to begin expelling it into the soil. And the bugs we've run around, I think the growth cycle for our radishes is around 4 weeks or 5 weeks, and we've run 6 crop cycles on them. And by the way, I'm not saying this is an industry that's ready to go next week, because of course I have, talk about GMOs, I have these weird little things in the ground, but I just wanted to find out, could we actually do it? And so is there a way to do this using the sun? So the answer is yes. There's a whole bunch of societal issues and everything else we'll have to deal with. But sometimes when you're doing science, you just want to prove that, get to the top of the mountain to prove it can be done.
Ben Valsler, Chemistry World. What sort of quantity or what mass of this do you need to put in the soil before it's producing enough ammonia to act as a fertilizer?
Geez, I don't have that answer. I could get you that answer, of course, because we know how many radishes we're growing and we know how many bugs we put in the soil. I will say it's not a lot. And so unfortunately, I'm going to give you a science answer, which I don't think you want. You want to just know how many bugs versus how much radishes that I make, and I could figure that out for you. But we do know we can measure the turnover number of a bug, so the how many times it takes nitrogen to ammonia, and that's 8 times 10 to the 9th, which is basically 800 trillion cycles we can run on one bug. And so that's why it's working so well. If you're doing nitrogen fixation in my field, you hope for a few turnover numbers, like the world records, they get published all the time, 1,500 turnover numbers. When we saw how much nitrogen we were making, we said, well, we should see if the rubber hits the road. That gave us this idea of building this parallel cycle of polyhydroxybutyrate and then burying them in the ground to see if crops grow. So it's making a lot of ammonia.
So it's Kath O'Driscoll from Chemistry and Industry magazine. Just to ask about why you chose that particular species of bacteria, and also how do you expect that would interact with the natural community of bacteria in the soil?
Those are all good questions. Yeah.
And also, I was interested in the fact that this bioplastic acts as a kind of an energy store. So does that mean that you could theoretically grow plants at different times from conventionally?
Okay, so there's 3 questions there. Why did we choose the bug? And we chose this bug because it has a natural nitrogenase. So it has this nitrogenase enzyme, which takes nitrogen to ammonia. It has a natural nitrogenase, which is very tolerant to oxygen. And there's different ways nature can make nitrogenases tolerant to oxygen. One is it kind of builds a wall around this enzyme because the enzyme is very sensitive to oxygen. So other chemists are trying to take just nitrogenase itself on an electrode to do what we've done, or just the enzyme itself. And then they're always battling this issue. If there's any oxygen around, it will kill the nitrogenase. So we chose your first question is, why did we choose this bug? And it's because it has a tolerant nitrogenase. And then your second question was, does it interact with other organisms? And then your third question was, I lost one of your questions.
It was to do with, because the bioplastic is like an energy store, and you said to put it under the ground, does that mean it will affect when you could grow plants?
Yeah. So then the second question is, we do know so this is more complicated, and I'm saying the ammonia comes out, and we don't think the ammonia is directly fertilizing the plants. And I'm not saying anything new to soil microbiologists. They'll say, of course, we know it's working with another microbe in the soil to take the ammonia actually to an oxidized form of nitrogen. And then that gets taken up by the plant. And we haven't identified what microbe in the soil its partner is. So it's a little bit more complicated, the fertilization cycle in the soil, which is quite common even when you put regular fertilizer like ammonia on ammonia or urea type fertilizers commercially. They usually work with other microbes in the ground to finally do the plant fertilization. One part of it is to solubilize the material so it can get taken up in water inside the plant. And we haven't identified that microbe, its partner. And then you're precisely right about this, because we've basically bottled sunlight in the form of this bioplastic. You could do fertilization whenever you want, at any time of the year. Yeah, that's right.
We have an online question.
Not online, just me. Katie Cottingham, American Chemical Society. I was just wondering, how does this fertilizer stack up to commercial fertilizers?
Right. So I don't think we're doing anything different than a commercial fertilizer, but you're touching on a more important issue. And, you know, a lot of people know me as the artificial leaf guy, the guy who split water to hydrogen and oxygen. And then I'm going to tell you, everything I did and now that I've done now with the commercial, this fertilizer version, is for our society not needed. So how does our society work? You build large industrial plants with distribution systems that cost trillions of dollars. And so even with water splitting, to make hydrogen, I split hydrogen from water. There's a thing called the water gas shift reaction where you can take H2O plus CO to H2 plus CO2 at large industrial scale. And then you can take the H2 with CO and synfuel, and you can do Fischer-Tropsch, and you can make lots of fuels. It's going to be a little bit more expensive, but we can do that, and you distribute. And we do Haber-Bosch the same way. So I've always felt the only importance of my research is, can I do what you do in this society in large industrial scales with distribution systems in your backyard for the poor? That's why I've always been focused on the poor. And so I don't think this fertilizer is any different, except that if you're a poor country in Africa, you don't need to convince somebody to build a Haber-Bosch plant, and you don't need all the money to build a distribution system. And I'll say that about any of our research. I don't think sometimes my colleagues understand that. We're really up against an infrastructure issue, and that's why I think most of the science they're doing, whether they realize it or not, are for the poor that don't have that infrastructure. Oh, and there are my big radishes. I didn't realize they were up there. Oh, look how big they are.
Hi, Doug Dollamore, American Chemical Society. Doctor, I'm wondering if this process that you're developing could be more tailor-made or for the individual crop setting than the commercial fertilizers that are available now.
Yeah, I think so. So what you're driving at is this. And this is a booming field that goes even beyond chemistry, this field, what's called synthetic biology. And so what you're doing is you're genetically encoding organisms with different enzymes and different processes. And so the answer is yes to your fertilizer question. And then I can go on, take one more step. I worked for Harry Gray at Caltech, who's, as you all know, quite a scientist and character and figure in our field. And Harry, I remember many years ago saying, someday the sun's going to do everything. It's going to manufacture everything. I mean, and he was just being more of a visionary, just saying, I think it can happen. Well, now I can see how it can happen, because if you think of what I'm saying is I split water to make hydrogen. So that's where I've done the solar piece. Then if this trick of using an enzyme, a hydrogenase, so the bugs only live off hydrogen, I could, in principle, genetically encode bugs with anything. I could make steroids if I put the right machinery in a bug to make drugs. I've talked to my student, Michelle Chang, a professor at Berkeley. Could she genetically encode, she's a synthetic biologist, to make starches? So, you know, The Martian, where he's trying to grow potatoes, well, you could imagine having bugs that take hydrogen in, and they're genetically encoded to make starch. And so I'm really excited by this idea that we're on, because I see it in a much broader sense as a total renewable chemical synthesis platform, and where you tailor whatever you want by the genetics inside the bug.
Thank you. Thinking about the practicalities of this bug, what are its tolerances like in terms of pH and alkali and salt levels, water, and so on in the soil?
Yeah. So in the soil, it's very happy, but there is a salt issue which is really important, and it's not for this bug that's making the fertilizer, but it's for them. You're splitting water to make hydrogen and oxygen, and so the more salt you have in there, the better that is for the hydrogen producing step, because you have a lower, what's called ohmic drop, or internal resistance. And if I have an internal resistance, I have to put more voltage into the system to drive it harder to get over the resistance, and that wastes energy. It wastes solar energy to make hydrogen. And so if you can increase the salt concentration, you decrease the resistance in the solution. And we are at salt concentrations that are natural, so that's why the bugs in the ground are happy. But for hydrogen splitting, we want to be at super high salt concentrations. So we are working on the bug that where you're making bugs that are tolerant to very, very high unnatural salt conditions. So as we're generating the hydrogen, if we're doing it in their presence, because you don't need to necessarily, I could make the hydrogen over there and pump it into the reactor. So, some of the configurations we've used, they're all in with the hydrogen as it's being made. Then we want really high salt concentrations. So we're working on that. But in terms of a natural surrounding, these bugs are really perfectly happy. Like I said, we've gone through several crop cycles and they've survived no problem until their polyhydroxybutyrate runs out.
Bela Buslig, ACS. How stable are these bugs? Like you normally grow bacteria to reproduce and just keep producing more bacteria.
Yes.
I've noticed in the description that there were some catalysts in there that are toxic, so which were replaced by some non-toxic ones. Well, the bacteria to survive as a construct would it need access to the cobalt and phosphorus and so forth?
Yeah. So this is how they grow, and so first bacteria, we're usually trying to get rid of them because they like growing so quickly, like you said. So we take advantage of that, and so the way it really works is we only throw a pinch of bacteria in some water, then we give them lots of hydrogen, and they grow, and just like you're saying, bacteria grow, they grow exponentially. And so that means they're taking all their hydrogen, and with CO2, they're going into biomass, and they'll just keep using the hydrogen to grow. So what we do then is adjust the hydrogen relative to the hydrogenases, how fast they're eating, and we go into what's called steady state conditions, where some die, but some live. Then we take all the extra hydrogen and divert it along these pathways we want. And so the beauty of the bacteria is that you don't need to start off with a lot. You just start off with a pinch and take advantage of what you're saying. Let them grow exponentially like bacteria like to do. Then we adjust the hydrogen so that we go into this steady state. Some are dying, some are living, but their overall concentration never change. We stop exponential growth. Then they use the hydrogen for everything else. In terms of stability, there's two types. Bacteria are pretty rugged, and even in the soil, that was one question that was asked. That part is good. There's another stability biologists worry about, and that is if you put lots of genetics into them, as they're dying and growing, there's a genetic back pressure to go back to who they were. And so you put all these genes in, and then they want to delete all the genes because they say this is unnatural, and they go to be who they are. So these bugs are both the one published in Science for Fuels and then this one that we're using now, we're not very far away from how they function, so there's no genetic back pressure. I answered a question before that I see this as a general chemical synthesis platform to make starches and everything else. There will be, I'm kind of throwing this out like this will be easy. The more and more you put the more genetics you put in, the more intolerant the bug gets to those gene manipulations and then starts wanting to go back to who they are. So that's one challenge of what I'm talking about that synthetic biologists spend a lot of time thinking about. And so there's another neat thing about what I've done. There's this whole world of synthetic biology working away to figure out all these problems, and then I can interface immediately to it now with solar water splitting through this trick of using hydrogenases. So I like it because you're building and interfacing to all this activity in the world scientifically that you can just then keep bringing into your systems to do what you need them to do.
We have time for one more question.
Sam Lemonick again. Can you describe what it actually looks like when you take the bugs and add them to your radish field? And then I'm wondering if there's a way, you talked about combining the two steps of water splitting and bug hydrogen fixing. Is there a way to close the whole thing up into one so that you have the bugs directly making the bioplastic in the soil, in the field,
Yeah.
and then releasing the...
I don't think so, Sam, because the bugs, once I go under the ground, they can't see the sun anymore. So that's why we really split this up, and that's how nature does it. It always splits it up. So the leaf is doing its thing with the sun only to split water, as I'll explain today. And then in the dark, it's taking H2 plus CO2 to biomass, and the same with these bugs. And the other thing is I wouldn't want to take a field and put hydrogen over it. So I've avoided that problem, that I don't have to put gases over the field because I've stored the hydrogen as a bioplastic. So I don't see a way around that right now. And I'm not sure if you would want to. As far as what they look like, it's just like you have these bugs. They're actually more or less colorless. It's turbid. And then we just spray the water solution right onto the ground, and that's it. We don't do anything else. It's as simple could be.
Thank you. The archived version of this session will soon be posted at bit.ly/ACSLive_San Francisco. Please join us for our next press conference today at 1 p.m. Pacific time about how sniffing urine to detect prostate cancer could prevent unnecessary biopsies. Thank you very much.